Welcome to The Long Run. This is a podcast for biotech adventurers. I'm your host, Luke Timmerman. Today's episode features a conversation with Katrine Bosley. She's the CEO of Cambridge, Massachusetts-based Editas Medicine. It's one of the young companies seeking to use CRISPR-based gene editing technologies to develop new medicines. Editas is still a very young company. It was founded in 2013 with a license to use the technology from the Broad Institute of MIT and Harvard. Within 2 years, the CRISPR-Cas9 technology has spread like wildfire because of its cheap, fast, easy, and rather precise way to edit out genes across the genome, including single genes implicated in serious diseases. In 2015, the company raised a big venture round with big names like Bill Gates, Google Ventures, now GV, Fidelity, T. Rowe Price, and more. And 6 months later, without any drug candidate yet in clinical trials, it went public. Almost every day, CRISPR is in the news, either for scientific or ethical reasons, or both. Before this episode was recorded, scientists reported successfully using CRISPR to edit a serious gene mutation out of human embryos. In this conversation, Bosley and I talked about some of the day-to-day management issues around working in such a high-profile field and some of the additional responsibilities that come with that territory. In some respects, Editas is a biotech company like any other. It must harness its science and rally its people around solving a clear medical problem. It can't do everything all at once. It must prioritize. But at the same time, it can't put on blinders and be oblivious to the wider world uses and debates around CRISPR. What happens outside Editas' walls and is beyond its control can have a big effect on its destiny. Bosley, a 25-year veteran of biotech, thinks widely and deeply about not just her company, but the societal issues as well. Before taking the Editas job, she spoke with George Church, the Harvard University genomics dynamo, extensively about the ethical implications of CRISPR. I think you'll enjoy hearing her perspective on how an entrepreneur can size up a situation like this and stay focused on the ultimate prize, a drug that matters. Now, before we get started, a couple quick plugs. If you like this show, I think you'll love my subscription newsletter, Timmerman Report. You can go to timmermanreport.com to subscribe. And if you like the show, please spread the word on your social networks. You can subscribe for free to the regular download on iTunes or Stitcher. And if you have some positive comments, please leave them there so that it will help others discover the show. And there will be plenty more to discover. The next episode will feature a conversation with George Yancopoulos, the president and CEO of Regeneron Pharmaceuticals. You won't want to miss that one. Now, join me for The Long Run. Welcome, Katrine Bosley. You're the CEO of Editas Medicine. Great to have you here on The Long Run.
Thanks, Luke. It's great to be here.
So Editas, in my mind, really kind of epitomizes one of these biotech companies that's playing the long game. You know, you're founded in 2014 or late 2013, maybe, on this idea of building on the recent developments of really quick, cheap, easy genome editing tool by the name of CRISPR-Cas9. And there was, it's kind of a classic startup story in some ways. A big technology idea, you got some big name Boston VCs around the table, intellectual property license from the Broad Institute with a couple of prominent scientists involved. But then the question becomes, how to build a company, how to make medicines off of this platform technology. And that's where someone like you comes into the picture. How did you arrive at this?
Well, as you might imagine, the world of biotechnology becomes a pretty small world if you've been hanging around in it for 25 or more years like I have. I've been involved with other young companies and new technologies in the past. And I had the opportunity to be looking for a new opportunity. And what you get to do is you get to talk to everybody about what's going on and what's exciting as well, what do people need help with? A lot of the ways that you become familiar with new opportunities is you just spend some time with them. You say, what challenges are you working on? And you work on problems together. With this particular area, I actually found out about it from multiple different angles. One aspect was I spent a few months as an entrepreneur in residence at the Broad Institute, which one of the founding scientists of Editas comes from the Broad. Feng Zhang is a scientist and a professor there. And I met him while I was an entrepreneur in residence, and we talked about this. We talked about all kinds of other things too. He's a very creative and prolific scientist. But, I heard a little bit about it there. But I also heard about it from, reading the scientific literature. I also heard about it from the venture capitalists who were involved in putting the company together in its earliest days. There were folks that I knew from prior companies. And I remember one particular meeting with Kevin Bitterman, who's one of the investors in Editas. And he was just sketching on the whiteboard what this was and in terms of the basic technology. And the ideas of building a company around this were really to say, can you take this very powerful basic science and translate it into medicines? And in particular, the really powerful idea behind it was, can you make medicines that repair broken genes? If you think about it, your DNA, the core of all of who you are as a person in some respects, we know that mistakes in DNA can lead to very serious genetic diseases like cystic fibrosis or sickle cell disease. There are forms of blindness that are driven by genetic mistakes. And we've known this basic concept ever since we've understood how DNA works. But being able to repair, at the level of DNA, repair those broken genes has something that's been really more science fiction than a real concept until pretty recently. There are a couple of younger gene editing technologies, but the emergence of CRISPR as a gene editing technology, which is really just the past 3, 4, 5 years, has really transformed the landscape and opened up this set of possibilities of treating genetic diseases at the root cause, at the level of DNA.
Yeah, I mean, I think the other technologies that you reference are probably the zinc finger technology and talons, which have been around for a while and have been useful tools for scientists. But I think they have been a little cumbersome or expensive or just difficult to translate into a therapeutic. Or it's been hard for people to imagine turning those into therapeutics. And CRISPR, with its speed and specificity and cheapness, all of that combined, it sort of took off like wildfire in academia and got people's imaginations going in a new way in terms of how you might make medicines.
I think that's right. I think that point of really driving people's imagination is an important one because it's easy to use, at least at a research level. It's still hard to make medicines, but using it at a research level, it is robust. You can pick it up and use it without being a gene editing expert. And so people who are scientists who are expert in many different kinds of biologies and diseases and targets could pick up this tool and apply it to that area of their biological expertise and ask and answer questions and probe biology. And that has, I think, been why you see it spread so broadly within research science, is that it's a robust tool. Many people have said to me, my first experiment worked with CRISPR. And that's unusual. Science is often a lot more finicky than that. And so the way that this has driven imagination and helped people reimagine what kinds of targets to work on and to discover new targets. It's also a great tool to discover targets that you might use to make therapeutics. So it's really changed the face of biological research dramatically.
So you got on board in 2014, and we should back up a bit that before this, you had been the CEO of a company called Avila Therapeutics, which had been acquired by Celgene, and it developed a covalent binder for the Bruton's tyrosine kinase, BTK, for the treatment of cancer. And so that gave you some of that flexibility, that time off to think about what your next move might be and to settle on this one.
Yes, and I think too, it's a great example as well of a company that had a broadly applicable platform because, the idea of making the kind of molecule we were making, which is called a covalent small molecule inhibitor, but that kind of molecule, there are lots of different disease targets you could approach with that chemistry. Interestingly, it was an idea that most people who work in medicinal chemistry thought was a terrible, terrible idea. And it was our scientific founder there, Jaswinder Singh, really had a vision and an inspiration that if you did it right, it could actually solve problems you couldn't solve with other chemical approaches. So, a very orthogonal thinker in many respects. And you had to kind of go up against a very deep-seated dogma about why it was a bad idea and address it with data, with science as to why actually it was a good idea that could solve unsolved problems. So fascinating as well because completely the opposite end of the spectrum from CRISPR science where everybody's excited and diving into the pool. My last company was one where everybody knew for sure it was a terrible idea and we had to overcome that dogma and that resistance.
Yeah, as the CEO of this company, Avila, you're facing a wholly different challenge, which is to, make people believe in it in the investor community and partners, as well as the internal things that you have to do to manage the company. But, and in this instance, maybe you need to cool things down a little bit at times because there's so much excitement, or manage the expectations. How do you think about these two different set of circumstances?
Well, the common thread between these two circumstances is the science is incredibly exciting. So it does not take much for me to get going on talking about the science because they do hold great promise, and in each case hold the promise to solve problems you couldn't solve with other approaches. And ultimately, that's the starting point for any of this is what kinds of medicines can you make, and defining a very careful path to get to those medicines. In the world of CRISPR, communication is something we think about every single day, partly because there's a lot of different really important audiences and stakeholders. First and foremost, the patients, of course, patients and their families, physicians who are treating those patients, regulators, and not just the FDA, but more broadly, given some of the social and public policy implications of this technology, there's broader communication that becomes important with legislators and folks in the administration, and not just in the US, but more broadly than that as well. In addition, you're right, there's a lot of heat and light on this subject, and part of what we want to do is, yes, convey our excitement, but be very sober and realistic, because at the other end of everything we say, there are patients who have great hopes for this technology, and we're very sensitive to what they and their families hear, so that we're being as responsible as we could possibly be in speaking to all these audiences, but knowing they're listening in particular all the time.
Yeah, it seems like an unusual spot to be in. I think most biotech companies are in the more obscure spot, a little more like Avila, where not that many people know what you're doing, not that many people understand it. And in the case of CRISPR, I mean, not a day goes by. I mean, it's in the news every single day. And these are external forces that can actually have an effect on a company like Editas or some of the others in your small group of CRISPR companies like Intellia and CRISPR Therapeutics, or a couple of others. I mean, I was just on vacation last week and saw that there was a report out in the scientific literature of germline editing of human embryos. And this is the kind of thing that gets people's imaginations going about, the ethical issues, the designer baby fears, concerns that have been around for decades. And if perceptions run wild, that could, create some kind of backlash that makes it more difficult for companies like Editas or Intellia.
Yeah, and I think one of the reasons that we think it's so important to be very proactive in communication and engagement, and to be very to mean it and be real with that is that it's not just about what we're trying to do as a company. You're right. You're pointing to these broader questions of ethics and public policy, and everybody's got a stake in that. So part of how we can be constructive is, one, to be transparent about what we're doing. And there's a lot of ways we do that. As a public company, we, of course, are required to talk a lot about what we're doing, and we're very comfortable doing that. But we publish scientifically, and so there's scientific engagement. But we engage with policymakers, and then I think also try to be a responsible voice to the public at large, and not just us, but the other companies in the field, CRISPR and Intellia as well. And the three of us we talk to one another about these points because we have a very shared set of values around this, as well as desire to be very thoughtful and responsible in this communication. It is, I think that we can't lose sight of the fact that it's also a fantastic opportunity to talk to the public about science, and for us to then be able to understand what people's questions and concerns are, help all of us separate fact from fiction. But there's a range of views on these technologies, not just in the U.S., but internationally. And everybody's got a stake in these sorts of ethical questions. No question about that.
Do you have a Google News alert to keep you apprised of everything that may be incoming?
I have many information sources. It's interesting because I think that being in the field, obviously we're quite close to the scientific advancements. And so we often do hear about things earlier than they get published and such. So we're oftentimes, by the time they are published, we already know the science. And then a lot of the amplification of that is just sort of retelling the story or the breakthrough. So there's certainly a lot to keep up with. It's a very fast moving field. But I think we try to also separate the signal from the noise, if you will, and really make sure we understand deeply those significant advances or significant issues, and then try not to get lost in the noise of all of it.
Yeah. Well, this is the public-facing part of things, but tell me about how you think about what's going on behind the scenes, how you take an exciting platform technology and build a real company around this. In terms, how do you think about the three legs of the stool that we talked about the other day: the science, the business, and the people that you want to put together here?
Yes, and so let's tackle them in that order. So let's start with the science. As you mentioned, when this company was founded in late 2013, some of the core science was fairly newly published. It hadn't even been translated into a lot of different laboratories, and so it was a pretty exciting but pretty raw idea. What we did also have, though, was a landscape where progress in some other closely related scientific fields were very useful. So, for example, gene therapy. A lot of progress in gene therapy delivery had been made over the last literally 20 years. And looking at that, you said, some of those approaches might be useful in delivering gene editing medicines. So that's interesting. Also, a lot of progress in the science of sequencing and how do you do DNA sequencing, and the speed and the accuracy and the cost of that and the improvements in that was an important underlying capability. So you looked at all that and you said, all right, what kinds of problems are we going to have to solve to translate this academic science? Number one, you have to make your edits to the DNA very efficiently. So how do we improve efficiency? Number two, we have to do it very specifically. And that actually raises two questions. One is, how do you make a very specific molecule? How do you optimize the molecule itself to make it very specific? But how do you know? Literally, how do you know if your molecule is specific? There's no off-the-shelf analytical technology for that question because people haven't been doing gene editing before. So there's also both bringing in and then creating new methods to assess or to analyze specificity. So that, optimizing your molecule is one dimension of what you have to tackle scientifically. So that lays out a certain set of scientific questions and problems to solve. Second, how are you going to deliver it? How are you going to get it to those cells in the body that you need to treat, where you need to edit the genes of those cells for the disease you want to treat? So delivery is a second domain of technology development and questions to answer. And then how are you going to make sure you're getting the right kind of repair? There are 6,000 different genetically driven diseases, and there's a range of kinds of DNA mistakes that can lead to those diseases. Different kinds of mistakes need different kinds of repairs. Some are easier, some are harder. So you take all of those questions and it starts to say, all right, these are the technological problems we need to solve. And it starts to tell you what kinds of people you need to hire. You need to hire people who understand how DNA is repaired, how the cell reacts to DNA. So DNA damage repair specialists, people who understand delivery, people who understand the chemistry of RNA and DNA. There's oligonucleotide chemists that are important expertise to have. People who understand protein chemistry. How can you engineer the protein? So you start to understand the different kinds of expertise you need. Obviously, people who understand gene sequencing and things like that. But you need to intersect all of that with what kinds of actual programs do you want to work on? What diseases? What targets? And you do a big, long exercise, and that's what the early folks in the company did, of looking at all the different genetic diseases and talking to experts in genetic diseases and getting ideas. And, there's lots of different criteria you're going to apply to think about what to work on first. But it boils down to a couple big categories. First of all, the medical need. What is the nature of the disease? How serious is it? Are there other treatments that work? If so, then that's great. But, probably we should focus on ones where there aren't any good treatments yet. Number two, technically, can you figure out how to approach it? Given the type of the mutation, do you understand how to edit that mutation? And given the cells in the body you need to reach, do you think you can get there? Some cells are easier to get to than others. Right now, reaching cells in the eye or in the liver or in the blood or the bone marrow, those are more approachable. Cells, other tissues in the body, those are more difficult. So it doesn't mean impossible, but just some are easier, some are harder. So thinking about the technical tractability, can I get the right kind of edit accomplished? Can I get to the cells I need to get to? And, then can I find the patients? Are they identifiable? Can I reach them in time, given the progress of their disease? So you look at these different factors, and there's no algorithm for it. I mean, these are all important factors, and ultimately it's a judgment with regard to what programs you work on. But through that kind of assessment, there generally are a few programs that emerge as ones that make sense as starting points. And so as an example, we're working on a disease called Leber congenital amaurosis 10, or LCA10. And this is a disease where a mistake in one gene leads to blindness. And it's understood what the mistake is and what the downstream consequences are, and there's no treatment for this particular disease. But we can reach the cells in the eye. The mutation was understood. There's a lot of reasons we said that's one we think we could begin to approach. Working on the technology through the lens of a specific program is incredibly helpful. I always use the phrase internally, it's a forcing function, right? A forcing function is one that makes you solve for X. And when you're working in a specific disease, you have to solve for things like, how much do I have to deliver? How many cells? Yeah.
This is a very important task of management, setting the priorities, deciding what you're going to focus on, especially with an enabling technology like CRISPR, which can do so many things. Because you are a company and you can't do everything under the sun. You do have to decide. And it's interesting to hear you talk about those selection criteria and how it helped you focus in on Leber congenital amaurosa type 10.
Well, but if I may add one more layer onto it, because you're right, it is a question. You have to pick and choose and focus, or if you try to do everything, you'll fail at everything. It's just, you really need to create points of focus. But one additional layer you try to think about with a broad platform like this is to say, how can, in addition to that first program, hopefully delivering an important medicine for those patients, the technical progress we make with that program, how can that also teach us things that we can leverage in other diseases? So, for example, figuring out how to deliver to the eye, there are other eye diseases where hopefully that will be useful. We won't be starting from square one again with the next eye disease we work on. Also, because that's our very first program, some of the most basic aspects like specificity. How do you analyze for specificity? Well, we had to invent some technologies to do that, but those are now part of the whole platform. So that first program creates in its wake capabilities that you can apply to other programs. And part of what you want to do in these early programs that you select is ones where you can see how there are other programs that will benefit from the progress you make in the first one.
You want to get some leverage there.
Definitely.
So you, I believe, announced that you were going to take a little more time in getting to the clinic. You're still preclinical.
Yes.
So this something like another maybe 6 months, I think you told investors that you were going to do, and I believe non-human primate studies.
Yes.
Why did you do that?
Well, so with this lead program, our initial goal had been to try to file an IND, which is the documents that you file with the FDA in order to be able to start testing in humans. We'd originally hoped to file that IND by the end of this year. We, a couple months ago, shifted that to the middle of next year. The program is still making excellent progress. But one of the many challenges of a very new technology like this is it's not just about the biology, it's about all the aspects of developing a medicine. And you have to manufacture your product candidate at the highest quality level because you're going to be putting it into human beings. And so we do know how to do that, but it's complex. And so we ran into a couple of issues there that caused a delay. So we've solved those issues, but unfortunately it did cause a timeline delay compared to what we've been working towards. So this is somewhat part and parcel in the nature of drug development. In this particular case, the issue wasn't because it was CRISPR specifically. It just related to one of the manufacturing inputs didn't achieve a quality measure. That does happen sometimes when you're in early drug development, whether it's gene editing or small molecules or antibodies. We don't like missing a goal for sure, but at the end of the day, it's really important to get it right. And so we'll continue to advance the program. The data are still looking very good and very supportive of advancing to the clinic. And we just need to keep going.
Well, I think it's a fair point. I mean, people sometimes in the stock market get disappointed, nervous when they see a milestone delay. But at the same time, when you're in a field like this, a new modality here with CRISPR, if you rush to the clinic and screw up in a pretty big way, that would be a much bigger deal than, say, a 6-month delay after having gotten all your ducks in a row and moving more methodically.
There's always a balance because, of course, we have a huge sense of urgency for the patients we hope to help. And just because this field is so exciting, we want to move fast. And it absolutely has to be balanced with rigor and getting the science right.
I mentioned the non-human primate studies. Not every company does that. Or I know that they can be expensive and difficult experiments to do. Why did you decide to incorporate that into your preclinical work?
In our case, one of the things about non-human primates is that the physical structure of their eye is pretty similar to a human eye, and there aren't many other preclinical species that have that characteristic. So we can learn things, and we do work in other species preclinically, but we felt it was important to do some work in the non-human primate because that's going to give us the best understanding of what we think it will be like when we are advancing into the clinic. So it really was pharmacology science. This is, you pick the species you work in preclinically, need to be driven by what's going to give you the best information to support your clinical development. And that was the case here.
Well, this is one of the vexing challenges of the industry is finding, more predictive models. If we have more predictive models, we can reduce that failure rate and make drug development a lot more efficient. We're not there yet all the way, but working on it. So, you're still in very early days there at Editas. It's hard to believe that the first CRISPR paper was only 5 years ago. You still have so long to go. You know this in clinical development, and there's so much speed and excitement and urgency around it. Do you actually need to find yourself pacing yourself or slowing yourself down or encouraging your team to do that as well?
So I know you run marathons, so you can appreciate the concept of pacing. And I've run a couple marathons. I run them much more slowly than you do. But yes, pacing absolutely matters. And there are times when there's tremendous momentum and that's great. But what really matters is getting across the finish line. And so, having some sense of what's too fast and what's too slow. It's what we spoke about a moment ago, that balance of the sense of urgency with getting it right. I don't know that there's a formula to that, but I think part of how you figure that out, one is just building the team overall with people who have experience and who've been around the block. They've seen the movie before. On our executive team, we have people who've put multiple drugs into the clinic, and our chief medical officers brought multiple programs from IND all the way to approval. So they kind of know what it looks like. Similarly, on our board of directors, which, I think for some folks, what does the board do? It feels a little bit like a black box. Well, the board's an incredibly important resource, is how I look at them first and foremost, for myself as well as for the company broadly. Not only in terms of, obviously a sounding board for the things we're working on, but for perspective and for the experiences that they bring to the table. So folks like, for example, Akshay Vaishnaw is on our board of directors, and he's the chief medical officer at Alnylam. He's been part of developing that whole platform, seeing multiple new molecules go into the clinic. So a very, very helpful experience that's analogous to what we're facing here at Editas. At Alnylam, they're developing RNAi, so it's a different kind of modality. But when they started as a company, it was brand new. And what diseases to work on, and how do you deliver it, and all the kinds of questions that we wrestle with here, he's wrestled with there. So part of how you get the pacing right is you bring people into the company in these different roles who know what it's going to take, so you can figure out when to push, and when to maybe step back a little bit and think harder.
You must look at things like the Alnylam experience, and you recognize that they went through their long lean years, right? When people sort of lost faith for a while that RNAi wasn't going to solve all these diseases right away, or it wasn't right around the corner. Delivery was harder than people thought. Do you try to brace yourself for kind of that inevitable letdown, at least in some people's minds?
There's a reason I use the marathon analogy. I mean, it's like, mile 10, mile 15, all these miles are long miles at some point. And yeah, absolutely. It's I think Alnylam is a really great analogy. It turns out John Maraganore, who's the CEO there, he actually hired me at Biogen over 20 years ago. So I've known him through that whole journey he's been on. And yeah, you have to be prepared for those moments, because it's not just that it's new technology, it's drug development. It's hard. There will almost certainly be setbacks, and you have to look at those as problems that need solutions and figure out how to solve them and keep going.
What's one of those challenges in front of you that may not be very widely appreciated? Is dosing and delivery really one of them?
Yeah, in fact, it's funny because I was going to start talking about pharmacokinetics, but the question of delivery, I think we've all known from the beginning of this field, was going to be one of the big challenges for the field, because there are so many different tissues in the body you might want to address, because there's so many different diseases, and different tissues are probably going to require different solutions. There'll be some where you get leverage from one solution to another, but it's not going to be I don't think it's going to be a single delivery solution. So that it is a complicated aspect of what we're doing. But, you mentioned the question of dosing, and, the concept of dose, pretty straightforward, right? How much do you give? What's the right amount? You can ask that concept with aspirin. We all know a dose of aspirin, or a dose of any kind of medication we take. But here, we actually have to sort of invent a new language around dosing, because it's not about, X number of milligrams or something like that. It's really a question of how many cells do you need to edit to get benefit for that patient? And then also, what happens to those cells? If you're editing something like a hematopoietic stem cell, so this is the cells in your bone marrow that make your blood system, and they, keep making new blood cells all the time. If you edit those cells, and you might want to do that, for example, if you wanted to try to treat sickle cell disease, those cells will keep making new blood cells. So if you edit the stem cells, the hematopoietic stem cells, that population will continue to make more and more edited cells. So if you're editing a stem population, it's quite a different pharmacokinetic equation, if you will, than if you were editing, for example, the cells in the eye, the ones that we're working on in our LCA10 program. There you're editing cells called photoreceptors. And those cells don't divide. They're just there in your retina. And so once you've edited them, you're done. And they don't turn over. They don't propagate. They don't. They just are what are called terminally differentiated cells. So in these two examples, hematopoietic stem cells or photoreceptors, your whole question of how much is enough has a different set of parameters. And so figuring out not only, what is the right framework to talk about this, but then defining specifically for this disease, what's the number? How many cells do I have to edit? How much of my gene editing machinery do I need to deliver to get that many cells edited?
Not only is it a question of how much, but how often. Do you need to do repeat dosing or not?
And that's going to be very dependent on the disease. Once you've edited a particular cell, that cell is not going to revert and become unedited. So once you edit a cell, it'll stay edited. So if it's a stem cell that makes more cells, all of those progeny cells will be edited as well. If in a single dose you can edit enough cells that gives the needed therapeutic benefit, then yeah, maybe once is enough. But if you weren't efficient enough in your first delivery, or if you thought, you got some benefit for the patient in your first delivery, but if you could edit more cells, you might be able to do better, then you might want to have more than one administration. So this does get back to that question of dose, really, and to say, all right, how efficient is my editing in a single administration? Can I get the maximum efficiency in that one administration? Or if there's some technical reason I can't do that, then would a second administration be able to get further editing and improve what the clinical benefit is for the patient?
Are there other companies besides Alnylam that you've looked at as sort of case studies for where you want to go with this long term?
Well, if you think about some of the great companies of our industry that really pioneered broad new science, there are always lessons you can learn. The context is always unique, but we can look at Genentech and Amgen and Biogen and Genetics Institute, the earliest companies or CETUS for that matter, the earliest companies in modern day biotechnology. I think there's lessons from all of those. What programs do you work on is one of the top ones. How do you create enough capital foundation so that you can weather the storm? I mean, these are lessons that I'm sure Alnylam learned from those earlier companies as well, and we see them again with Alnylam. But some of these are are quintessential for the industry. I think, though, I in addition to a company like Alnylam, I think it's very interesting to look at the antibody companies, right? So you think about, you had the earliest antibody approved as a drug, OKT3, was a murine antibody, a mouse antibody. And then it was years before the next antibody was approved, because technically a mouse antibody is not a good idea as a human drug. And there was a lot of technology development that had to happen to get to what ultimately was the promise of antibodies. And you had protein design labs and it created the CDR grafting technology, which was a step that really allowed you to finally make lots of antibody products. And then you had other companies like Medarex and Abgenix that had even better technologies. So you could see that
there was a long effort to humanize the mouse, to reduce the mouse DNA and to make these antibodies less likely to provoke an immune response.
Yes, and similarly, less kind of visible. But the other improvement in technology that was happening at the same time was manufacturing yield and capacity and stability and a lot of aspects about the physical product that also was necessary to really start to address these larger patient populations. So you had this multi-year period before the field broke open, and there were a very small number of companies that were at the crux of really solving those problems. And then you had a lot of other companies decide that they were ready to be part of that field. So I think one of the questions I look at in the antibody field as an interesting lesson is, what is that set of technical capabilities that, and it's never just one problem you got to solve, it's usually a few, that really allow the full vision to be realized. And with antibodies, it was humanization and it was manufacturing and stability a little bit. So, I think that's one where I don't think we can yet see exactly what the answer is for gene editing, and we have our views and our thoughts. But I think there's some really interesting lessons from the world of antibodies.
What about hiring? Different kinds of people at different kinds of stages of the company. You mentioned this a little bit earlier, that you look first at the scientific problems that you're trying to solve and use that as your guide. But how else do you sort through the resumes and determine who to hire?
Hiring is probably the number one thing that I think about, hiring as well as team and organizational development. It's just never easy, because given the fact that we're in a new field, and so every single week there is something that comes up in some aspect of the company where the answer is, well, not only have we never done that before, nobody's done that before. So that in and of itself tells you need people who can think creatively, who are problem solvers, who figure out how to adapt ideas from other areas and solve problems in this area. People need a real resilience for that, because there are some weeks where it's like, hey, could we have a week where just nothing dramatically new happens and we just make progress, We do not have any boring weeks here, that's for sure. But I do think you really do need people who are comfortable in what can sometimes be an uncomfortable landscape, because there's just always something new that has to be sorted out. You also need people who are comfortable where the organization is not yet fully formed. We're a little over 100 people now, and we were half this size a year ago, and a year before that we were half that size. And that pace of growth means that we need to change as an organization as quickly as the science changes. And that, that requires a certain patience, but also people who are willing to identify what we need to do differently. Hey, we used to do this informally. We need a little bit more of a process now. And sometimes process is your friend, and so I think people who are sensitive to figuring out what the organization needs, not just what the science needs, but what the organization needs, because that's a constantly moving target as well. On that front, there's obviously tons of literature and experts about rapidly growing organizations, and that stuff is actually helpful. That's an area where we don't have to reinvent the wheel as much as we have to reinvent the wheel on the scientific side. So I think part of it is also getting good help in terms of how do you do the organizational development? And then finally, just hiring, you're always talking to people to get ideas for the people that might help contribute to the organization, both in terms of employees, but also developing the board of directors, because that's another dimension of developing the organization.
Well, some of this is a natural evolution, right? As your company evolves, so at some point next year, you'll be in the clinic and you'll have more requirements for, say, regulatory people than you have now. And these things will just, clinical development, et cetera, they will layer upon each other. The whole thing expands. But I guess you mentioned process. That's often kind of a dirty word to a lot of people in the industry. When you're trying to do something is, adaptable and at an adaptable stage, you don't want to become a prisoner of the process. But I don't know. I mean, at this stage, 100 people, it's not that much of a danger that everybody will be in their little silos.
No, but I think, so two things though. One is that, process can be empowering, right? If everybody knows how it works, then you don't have to have a meeting all the time to figure out how it works. People are empowered. So I think figuring out what processes are actually empowering rather than bureaucratic is part of the key. But the other thing, so yeah, you're right. Some of it is naturally as the company scales, we'll need more capacity in certain roles. But one of the things in these early years of a company that I think is critical is, in any given function, the first person you hire into that role has a profound impact on that dimension of the company writ large. It lasts. Like, that individual's impact is, it's the first regulatory person in the company, the first clinical person in the company, the first communications person in the company. These individuals really set a tone that becomes part of the DNA of the organization. And so I think that, sure, there's a natural scaling, but I do think you need to pay particular attention when you're hiring the first person in a particular function, whatever level that is. It really matters. That's
a really interesting insight. And I think that expresses some humility on your own part. Whereas, as a CEO, it's not all about you.
If we wanted to spend time on how much I don't know, you'd need a longer podcast. So.
I mean, but obviously setting a tone, a culture starts at the top. It's very important. But that's not the end of the story. What you're saying is that these people in their own domains, they leave very important fingerprints. Well,
and you need them to, right? Because, again, we said earlier that every week we face some challenge that's like brand new and pretty big and has a lot of impact. And for sure, you could never do this if stuff had to go through me. You need people in each role in the company who are far better at what they do than I would ever be if I tried, right? I mean, that's the whole point, right? This is a team sport. This is not making new medicines is not an individual sport. It's the ultimate team sport. And it extends to every single role in the company.
Do you think the timelines for what you're trying to do at Editas are pretty predictable in terms of the long term? Like if you look at something like Alnylam or Genentech, these things took a long time. Or do you think we're entering a different phase when maybe these timelines will be compressed and accelerated? Maybe we'll have a drug for Leber in 4 or 5 years.
Well, there's always some aspect of it that's a little bit disease specific, right? That the time it takes to study a disease like Alzheimer's, given the nature of that disease progression, is always going to be different than for something where it's a rapidly progressing disease. So there's some inherent difference in timelines based on that. But I think just stepping back and looking at, across the landscape of the last 40 years of biotech, what does it really take to build a sustainable company? It is a long endeavor, well over a 10-year endeavor, no question about that. But is it 20 years? Is it we still see these, we still know the companies that have kind of gone the whole journey and obviously continue to be vibrant companies. Look at a Gilead or a Biogen or I mean, Genentech is part of Roche now, but was independent for many, many years. Amgen, obviously, is a huge company. There's Regeneron. We could go on and on with different examples, some of the newer ones, Alnylam, but more recently, look at Agios. They've just had their first product approval, and that was fairly rapid for a company that was, I think they're 7 or 8 years old. So it can be rapid in certain instances, but I think that the way I'd always look at it is, plan for winter. Maybe it'll be a short winter, but plan for a long winter, and be ready for it to be quicker. I mean, you always want it to be quicker if you possibly could, but you always have to be prepared for it to be tough and long, and then work to find ways to make it quicker.
Well, and even in the case of something like an Agios, they've now got one drug on the market, and that's great. But to be, a truly enduring drug development enterprise, you need some diversification, like some of those organizations you mentioned, and that's going to take a few more years. It just does.
But it's funny. Yeah, I remember when Amgen was launching their third product. It's going back a few years now. Their first 2 products were huge blockbuster products, and their third product was important medically, but just wasn't as big. And they got beaten up because their third product wasn't as big. And I remember thinking, come on, that's their third product. That should be fantastic. So, I think it's one of those things that sticks in my mind as, there's always another aspect of building a company, and making drugs, no matter how big or small you are, how young or old you are as a company, making drugs is really, really hard.
Well, the market can be a very unforgiving place. It's necessary. The investors have to see that blue sky, that opportunity, and folks like yourself need to keep them informed and on an even keel as best you can, I guess, to buckle up for the bumpy ride.
Well, and I think part of the way we approach that is to try to be really rigorous and clear and transparent in our communications. I mean, we speak to investors all the time. They're a really important stakeholder for us. We're able to build this company because they believe in us and want to be part of that journey. And I think that, we want to be clear with them about the commitment that we have and the commitment we think it takes to really do this. And, I think there are absolutely investors who understand that. There's, obviously many different kinds of investors with different kinds of investment goals. And there are ones who really understand the long-term potential. And, we're privileged to have a lot of them as investors in this company. And we need to earn their capital just as we need to earn the trust of the public in what we're doing and everything else. It is an important part of building a company like this.
Yeah. I think this is part of what separates the really best companies from the mediocre ones, is that ability to communicate in a fair, responsible, even-handed, clear way. I think of back to at the beginning of the show, we talked about a 6-month delay on your timelines, and or there was a paper recently by some scientists that talked about off-target effects of CRISPR, lack of specificity in some instances. And that causes some people to get jittery and nervous and run for the hills. But that's not really news to the people who have been paying attention for some time. And it shouldn't be a 10-alarm fire. It's, and if you've done your job as a company, there might be a little chop in the sea for a while, but things will balance out over things like that.
Well, and also I think, how do you look at those as opportunities? So, for example, the paper, the short paper that was published in May, that asked questions about specificity, it did definitely cause chop in the marketplace. And we basically said, all right, well, we need to respond to it scientifically. We'll do that. But what a fantastic opportunity to talk to people about specificity, because we think it's hugely important. And it's always been part of our platform. And, a lot of times people don't want to hear about it because they think it's this little bit arcane technical detail. And we're like, we love talking about specificity. We think it's hugely important. We have a lot of we've got a lot of technology around that. We can address this question very robustly. So in a weird way, you never like to see your stock price get thrown around for essentially arbitrary reasons. But on the other hand, there were a lot of conversations we had with investors around that paper, because all of a sudden they were really interested in specificity. And we said, fantastic, let's talk about it. So I do think that you have to look at it that way, because we understand people having anxiety about, is this an issue or not? Great, let's talk about it.
Yeah, and this is the kind of anxiety that, it gives people heartburn in the short term. But, if you could fast forward, a year or so, and you've got your first batch of data from Liebers, and it's good. That's the sort of thing, like that paper will be long forgotten. Less than a footnote in the history of Editas, if you deliver on your goals.
Absolutely. But I think, we always have to answer with data, right? Whether it's a question somebody else raises that it is a real substantive question, or it's maybe people think it's related, but it's not, or whatever it is, our strongest answer always has to be with data. And I think you're right that, as this technology goes through all of its terrible twos and its adolescence and all of that, as we work out how to really make medicines based on CRISPR, ultimately what lasts is the medicines. And I think that the Sturm und Drang of how you get there, which is inevitable for new science like this, its lasting legacy is medicines.
Katrine Bosley, thanks very much for joining me on this episode of The Long Run.
Thank you so much.
Thanks for listening to The Long Run, a production of Timmerman Report. Music for the show comes from D.A. Wallach. Pedro Rosado of Headstepper Media was the producer and editor. Todd Bennings created the logo. Next on The Long Run, I'll be talking with George Yancopoulos, the president and CEO at Regeneron Pharmaceuticals. Regeneron, as many listeners of this show know, has been one of biotech's big successes of the past decade. It is the third best performing stock in the S&P 500 over the past decade. Yancopoulos and chairman and CEO Len Schleifer have been there since the obscure early days and have seen it all. They have strong opinions about the kind of culture they wanted to create at their company, on the right way to price their drugs, and how they want their company to continue to remain creative, or at least avoid getting stale. Don't miss this upcoming episode of The Long Run.